So today I'd like to talk about the human gut microbiome, their evolution within healthy individuals. And I'll start with a little bit of background. So we know that microbiome interact with our host environments, and together they regulate human health. But little is known how does the host environment drives the microbial evolution, especially think about the gut microbe has been evolving and colonizing a gut environment for millions of years. So are they still actively adapting, or have they reached the fitness plateau? And there are many studies reported pathogen species are adapting actively within the host after they invade into a host. But for commensal species, there's no consistent conclusions. In one study earlier in our lab, we showed that the B. fragilis are adapting actively within healthy human individuals. But there are also other studies prove that long-term purified selection is very common for gut commensals. But of each and every those studies usually focus on one species at a time. And as Professor Anni Zhang just mentioned before, the microbial host interactions are way more complicated than single species and host interactions. So in this study, we would like to look at a global investigation into the whole microbiome community and how they interact with host. And we would like to look at the selective pressure in the human gut microbiome, human gut that drives this microbial evolution. And I will go briefly through the methodology. And there are a lot of details here, and I would like to talk about the details later on in the Q&A session. But the most important thing here is that we try to look at mutations that happen recently within each and every person, and we want to differentiate that mutations from the ancestral mutations that happened like thousands of years ago. So to do that, we first define a lineage as a group of strains that potentially evolved from a single ancestor strain that colonizes a human subject. So all the mutations finding in these lineages are potentially happened within this human subject. So we first cluster isolates into lineages, and we find mutations or SNPs within each lineage. And then to differentiate genes under positive selection from genes under purifying selection, we look for a group of genes called PE genes, genes under parallel evolution. And they're a group of genes with multiple mutations that they accumulate independently in different strains. So here there are two examples of two genes with 4 mutations. And for example, this gene, the 4 mutations arise independently in 4 strains, and we would consider it as a PE gene. But for this one, this gene, all 4 mutations are kind of linked together on inside a single strain, and we will not consider it as a PE gene. And usually PE genes are found to accumulate non-synonymous mutations, and so they're proving to under positive selection in many species as an indicator for recent adaptation. And as to the data set we use in this study, the first data set is a public data set published by our lab a few years ago. We isolated 64 species from 15 healthy individuals. And another data set, we collected 3 species, the 2 Bifidobacterium and 1 Parabacteroides from 82 healthy individuals. As to the results, first of all, we find adaptive evolution is very common among gut commensal species. And this observation is supported by 3 lines of evidence. So the first thing we would like to know is, okay, whether the PE genes finding in our data are also under a positive selection or adaptation. So we computed the DNDS of non-PE genes compared to the DNDS of PE genes, and the PE genes were under a very strong positive selection with the DNDS of 2.2. And the second question is that whether our PE genes identified in our study is true positive. Because imagine you have a lineage with 10 SNPs. They're randomly distributed on 10 genes. It's highly likely you just get a gene with 2 mutations just by luck, by chance. So here we build a neutral model with 1,000 times simulation that assume that there's no difference of selection across genes and all mutations are random. And what would you observe? And those PE genes are probably false positive. So here in the x-axis, we display the mean of 1,000 simulation for each lineage and the 95% confidence interval. And y-axis is the observed number of PE genes. And we can see that most of the lineages are much higher than the diagonal line, which means they accumulate significantly more observed PE genes than expected. And this covers 24 of 27 species with sufficient sample size. And third question we would like to ask is, except for PE genes, is there any other adaptation traits we can see in our data? So we look for truncations, which is a type of mutation that causes early stop codon. And truncations are known to be adaptive, like a trait of adaptations, because they're very likely to change the protein functions. And we can see the truncations are highly enriched or significantly enriched on PE genes in our data. We also look for the metagenomes of a human gut microbiome in like Asian population, European population, American population. We also see the same trend that truncations are enriched in PE genes. So together with 3 lines of evidence, we can say that we observe widespread adaptive evolution among gut commensal species. And then we ask a question that what genetic pathways are the targets of adaptive evolution? So here we look for pathways with an enrichment of PE genes. Here each row is a pathway, and we computed the effective size of PE genes, which is observed divided by the expected PE genes. And here, for expected PE genes, we did 1,000 times of simulation. We assume that there's no difference of selection among all the pathways. How much would you observe? And then we find out the transcription and signal transduction, those 2 pathways, show significantly higher enrichment of PE than expected. And so those 2 pathways might be like generally under a strong positive selection. So which means there is positive selection on both PE gene and potentially non-PE gene. So we look for the DNDS of non-PE genes in all those pathways, and we find out that yes, transcription factor and signal transduction, they show a high DNDS compared to the average. And especially signal transduction shows significantly higher DNDS of non-PE genes. So together, we think that signal transduction show the strongest signal of adaptation. And because time is very limited for me today, so I would like to share the most interesting story that we find, most interesting PE function is this lactide HTH-type regulator, is msmR gene. It was under widespread adaptation in 7 different species, including several Bifidobacterium and Bacteroides. And they are known to regulate multiple sugar transporters. And we can see that here is a protein structure of this transfer factor, and here is a ligand binding site and DNA binding site and double strand DNA. And we can see there are a few variants right here at the DNA recognition site of this protein. And there are also a few variants or mutations right at the ligand site of the protein. And more interestingly, we find out that if we look at the different DNDS of different regions on this gene, for example, the DNA binding region, the ligand binding region, and other region, we can see that the domain region show much higher DNDS of 6 to 9 compared to the other regions that are non-domain, which show a DNDS of only 0.9. So in conclusion, we observe widespread adaptive evolution in gut commensals. It's supported by 3 lines of evidence, and we resolve the previous conflicts of previous studies. And signal transduction pathway show the strongest signal of adaptive evolution. And we find adaptive SNPs were significantly enriched on the domain region of this transcription factor, and we are doing experimental validation to see which carbohydrates respond to which adaptive SNP. And I would like to thank my husband Jeff and all my beloved lab mates, and Tammy and Jesse is my collaborator, and Erica is my supervisor for their guidance and help and support all the time. And thank you so much for your attention.